The human heart is unique among mammals for this key reason. ME drained from conversation reports, mammals, from the mighty blue whale to the tiny shrew, inhabit nearly every corner of our planet. Their remarkable adaptability to different environments has long fascinated scientists, with each species developing unique traits to survive and thrive. Despite such vast biological diversity, it was believed until recently that the structure and function of the heart across mammals was the same, but research from my colleagues and I reveals that the human heart is an outlier distinctly different from those of our closest relatives, the great apes, including chimpanzees, bonobos, orangutans, and gorillas. So why are humans the odd ones out? Humans diverged from chimpanzees, troglodytes, our last common ancestor, the theory goes, between 5 and 6 million years ago. In contrast, people evolved to stand upright, to engage in greater amounts of activity such as persistence hunting, and we have developed considerably larger brains. These changes to human bodies were associated with much greater metabolic demand, requiring more blood to be pumped into our muscles and brain. Our research suggests that the human heart is adapted to support our upright stance, movement, and larger brain. Over the last 10 years, we have been conducting assessment of the cardiovascular system of great apes across the globe. We've been fortunate enough to work with dedicated veterinarians and care staff in the UK, Europe, Asia, and Africa. An important aspect of these assessments has been the use of cardiac ultrasound, through which we can assess the structure and function of the heart, how large it is, and how the muscle contracts, twists, and rotates. Our previous research has suggested that the structure of the human heart may be different from that of the chimpanzee. Using cardiac ultrasound, we discovered that the left ventricle, the heart's main pumping chamber, in chimpanzees contains bundles of muscle arranged in a mesh known as trabeculations. For the current study, we wanted to examine if trabeculations exist in other great apes, which we found that they do. And in contrast, humans have a smooth wall of the left ventricle. This difference is especially pronounced in the bottom of the left ventricle, when the human heart's smoothness is nearly 4 times greater than that of our great ape relatives. Our research did not just reveal structural differences in the human left ventricle compared to that of the great apes, we also uncovered an important difference in function. By using a specialized technique called speckle tracking, ethnoechocardiography, which tracks heart muscle movement through contraction and relaxation, we examined how the muscle thickens, twists, rotates, and lengthens. The results were striking. Humans who have the least trabeculation exhibited much greater twist and rotation in the apex, the tip of the heart, during contraction. In contrast, non-human great apes with their heavily trabeculated heart shows much less movement. We believe the human heart evolved away from the trabeculated structure seen in other great apes to enhance its ability to twist and contract more efficiently. This increased twisting motion, along with the smooth ventricular walls, probably allows the human heart to pump a larger volume of blood with each beat. And this means that the heightened demands of our physical activity in larger brains, our research challenges the assumption that the heart structure is uniform across mammals. Instead, subtle but crucial differences in heart anatomy and function have emerged in response to unique environmental changes. Cardiac disease. While our current research has shed light on the evolution of the human heart, our work analyzing the hearts of endangered great apes continues. Sadly, cardiac disease is the leading cause of death in captive great apes. Unlike humans, great apes do not appear to develop coronary artery disease. Rather, their heart muscle undergoes a fibrotic or thickening process which causes poor contraction and the susceptibility to arrhythmia, which is a problem with the rhythm of the heartbeat. The cause of the disease is unknown, so we are at the International Primate Heart Project have been conducting assessments of great ape cardiovascular physiology across the globe to further understand the disease. Before our involvement, little was known about the normal cardiovascular physiology of great apes. Through collaboration with veterinary practitioners, our research has gathered vital data, significantly improving our understanding of human heart evolution, as well as the understanding, diagnosis, and management of heart disease in great apes. This is from the conversation under Creative Commons by Amy Drain, Senior Lecturer and Clinical Academic in Faculty of Medicine, Heart and Life Science, Swansea University of the UK, on Science Alert. Please leave your comments. Thank you for your support. Please support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.